Okay, we're going to continue our discussion about the regulation of gene expression. So this is the second part of that discussion. We've really moved into what will be a very complex area to talk about. So this lecture is going to take a little bit more time, even to cover, just to skim the surface of what's going on here at the level of transcription. It's going to take a bit of effort to talk about. This could be easily, in fact, is a whole semester course at more advanced levels where we talk about transcriptional control. So I want to go over some of the main points, though, so that we kind of understand the process of how transcription can be regulated and all the elements that often come together. Now, just to start, we need to think about the fact that, transcription itself and then the regulation of that is not a trivial process. There are a large number of steps that have to come on. While it is on-off type of system, it's not a simple on-off type of system. There's a huge amount of activity that must go on in the cell in order for it to happen. First, you need some kind of signaling function. That can be hormones, it can be growth structures, it can be nutrition coming into the cell, it can be sunlight, depending on what kind of cells you're talking about. It can be various things that occur, but you need some kind of signal. That can be either external to the cell, therefore in the form of hormones or some external input, or it can be internal. When the cell calls for the production of proteins itself, it will signal that. And cell signaling is one of the really important areas of research in molecular genetics. So once it signals, though, there's a cascade, it's called. A group of things have to respond, okay? And that's part of a cascade. Ultimately, what that cascade will do is activate a transcription factor. That transcription factor will bind somewhere onto the DNA, either in a promoter region, a TATA box, or maybe even at an enhancer. But that transcription factor will find its place and will activate. And we'll talk more about transcription factors in a moment. Once the transcription factors are in place, when they bring in all these other members of the transcriptional complex we talked about when we talked about transcription, there are the TATA binding proteins, there are these capping proteins, there are all sorts of things that come into play. Ultimately, once that complex is in place and in the right place, it starts to recruit RNA polymerase, and RNA polymerase moves into what we think of as the promoter region. And ultimately, once it's in the promoter region, you can start the process of transcription. So all these things have to happen. They're all controlled in various ways themselves. So there's actually regulation of each of these steps by the way they're methylated or the way the proteins themselves are formed into their tertiary or quaternary structures. All sorts of things can happen to this. Okay. Just to give one example of this, let's talk in general about maybe things like hormones and how hormone might work, because this would be one of the signaling components that we would need to think about. And there are lots of hormones out there. And we're talking about primarily here in mammalian systems. There's glucocorticoids, things like testosterone, estrogens. All these are things. Well, basically, what happens is that hormones are produced somewhere in the body. Okay, or if you're a plant or something, they are an insect. They can be pheromones. They can be produced externally and received. Okay, and in fact, we have some pheromone type receptors as well. But we're talking about things that are produced maybe in the testes or the ovaries or in the adrenal glands or some other area. These are small proteins. They're matured as proteins put out into either the blood system, a circulatory system, or maybe the lymphatic system, move around, but eventually they come in contact with the cells that they're supposed to contact. Those cells, in order to respond, must have receptors. Receptors are other proteins that can capture these and respond to these hormones. So these proteins have to work together. We'll see a model of that in just a moment. But the point here is that you have these hormones and you must have some type of receptor. Basic idea is that you don't have a receptor, you get no response. So this is one of the ways that we can produce a hormone in one area that will control tissues or cells in completely different areas while ignoring others. You might produce a hormone that causes a response in skin cells, yet it completely bypasses neural cells or something else. Because those neural cells don't produce the protein, the receptor protein, and therefore they get no response. This is also important for things, discussions that people have One of the big things is bovine growth hormone. We can synthetically make this growth hormone. It's given to cattle, milk cattle in particular, to make them produce large amounts of milk. And it's been banned in many countries. In fact, some of our milk that's produced in the U.S. is banned because we give it to the cows. And people, it's banned. Probably, it's a good thing that it's probably banned. It's banned for the wrong reason in many cases. Okay. Originally, what the concern was is that we were giving this growth hormone to cattle. They were producing milk, and potentially, as we fed that milk either to ourselves or particularly to infants, that they would have this growth hormone. It would cause this, uncontrolled growth, or maybe cancers, or all sorts of things that could occur. People were very concerned about, feeding this bovine hormone milk to children. The problem with that is that we don't have the receptor for that particular hormone. You can basically eat it, and it's not going to do much effect. Now, of course, obviously, anything in huge amounts will cause problems, but we don't have the receptors. We don't have bovine receptors in humans, so we're not going to. The hormone present of the hormone has no effect. Just like there's all kinds of, any room you're in, there are insect pheromones that are flying around constantly. Well, we're not responding to those in any real way. So, what's the difference? And there's really not much of that bovine hormone that actually ends up in the milk anyway. So, have no effect. But people are very concerned about it, and it's banned in many places. But we don't have the receptors, so it's really a moot point from that standpoint. It's not a moot point from what other things that it causes when you treat animals this way, particularly cows. In this case, you have other responses. You have IGF proteins that are produced that do end up in the milk that can be increased numbers in humans. We can absorb that. Probably more importantly, though, these cattle develop infections quite frequently because of so much production. So we have to give them lots of antibiotics, and those antibiotics definitely end up in the milk. And then they're fed to infants and things. We have problems from that. So you don't want to do that. Okay, you don't want to give a lot of antibiotics to anybody. You certainly don't want to do it through their normal food channels. And so, the bovine production of the given the hormones irrelevant. Okay, we don't have the receptors. The side products of that are things though that you do have to think about and probably are important. So we probably did the right thing for the wrong reason in sort of reducing the production of that type of milk in this country, and as well as others. Anyway, if we think about some examples though, let's take a fairly simple example that's not simple after you think about it a little bit. Let's talk about estrogen and how that might work, and talk about this concept of receptors. So we have estrogen hormones, okay? These hormones are made primarily in ovaries, so by females and mammals, okay? But they're also produced in the adrenal glands as well as in adipose and fat tissue. They can produce these hormones. The estrogen controls lots of different things. Obviously, it's important for the formation of uterine systems. It's for the menstrual cycle production. It's involved heavily in that, but it's also involved in the development of females. That lineage when we go off to be male or female during fetal development, and certainly secondary female characteristics, hair patterns, adipose disposition patterns, and those things are strongly influenced by estrogen. It affects cell cycle proteins, heat shock proteins. It's involved in the formation of bone and the restructuring of bone and calcium absorption, and it's actually involved with skin proteins as well. It turns out that thickening of the skin, and with estrogen present, you get the skin changes structure, etc. Okay. Now, the way it works is simply that it's a hormone produced in these areas. If there is a receptor in the cells, the hormone will combine with that receptor. It will be usually moved across the outer membrane of the cell, and you will induce this cascade of effects that eventually affects these transcription factors that are found either somewhere on the DNA, either in an enhancer region, depending on the cells, or close by to the actual gene itself. Okay. Now, where these transcription factors are and where these receptors are depends on what tissue type you're talking about. Estrogen does have an effect on things like neural cells. So neural cells have receptors for estrogen. Skin cells have receptors for estrogen. Obviously, the reproductive tissues have receptors for estrogen, but other things don't. Okay, so it just depends. And as sort of a side note of this, okay, what happens? Well, if you reduce estrogen, such as when females go into menopause, so in their 50s, usually somewhere in 40s or 50s, females will start to reduce the amount of estrogen they produce. This often leads to things like osteoporosis, because estrogen is involved in two things. One, in the formation of bone, as well as it's key for absorption of calcium in the gut. That's one of the places we get, obviously, from our food is where we get calcium. So if we can no longer absorb it from the gut, we tend to take, when we need calcium, we pull it from the bones, right? So that makes brittle bones. So it's one of the reasons we see osteoporosis occurring in these females. Another thing is once you have estrogen go down, the thickness of the skin starts to decrease, and the plumpness of the skin, the ability of those cells to retain water and these sorts of things. So part of aging process, this wrinkling and things that some of us that are older get, is due, in fact, to estrogen reduction. So it's one of the things that thoughts that we can do is we can put back estrogen, we can reduce the amount of osteoporosis, we can also keep some youthful look to the skin and more complex look to the skin. The problem with that is that tissues like breast tissue are very sensitive to estrogen and have lots of estrogen receptors. If you continue to provide estrogen therapy and provide estrogen to women who are postmenopausal, it works, but there's very clear evidence that after a few years, if you shut down that estrogen, then you stop the therapy, there's a fairly quick response that you see a higher spike in breast cancer. In fact, other types of cancers as well, but breast cancer being the most notable one. So, back in the 1960s and so, women would they would either have go into menopause, or they would have issues and they would have hysterectomies, or they just go through menopause. They would give them estrogen therapy, and it would just continue for long periods of time because it was preventing osteoporosis and doing other things. There's mood issues that go with it. Lowering estrogen actually has a neural effect and can be associated with some depression. So it was a good thing. The problem is when you stopped, then the spike, in the probability of breast cancer just goes up dramatically. So nowadays, when you go through estrogen therapies, there's a very short window of time that they'll actually provide that for women. Other things, what's interesting is that when you increase the estrogen, melanin production. Remember back when we did that discussion back about why you have chocolate labs and yellow labs and all that. And we talked about the melanin, eumelanin cycle, the split between those two. Well, melanophores don't actually have receptors, but they do respond somehow to estrogen. And so you actually see that when there's estrogens higher, you'll see an increase in the darkening of the skin, a more tanning capability. And it's interesting, women that take oral contraceptives, a lot of the oral contraceptives have estrogen and proestrogen is mixed in it, but estrogen's available, and they'll actually, you can tell that they are in the sun, their nose and other areas will actually tan more than when they're not taking it. So it's kind of an interesting outcome. And then in males, we do produce estrogen. It's in part both from the testes. There's a small amount produced there, but also adipose is very common is one of the areas. And it leads to a really important problem because fat cells, as we get more and more fat cells, we produce more estrogen as males, and it counteracts the other effects of testosterone. And so males that are really obese, that have lots of fat, lots of adipose around their body, actually have lower sperm count. And it's due to the fact and other characteristics that are actually due to this increased estrogen. So not only being obese an issue for health, it's also a reproductive issue. Okay? All right, so we've got estrogen, we've got going on. You can see that it causes this one little hormone, this one little protein produced in various places has massive effects. And what was the term we used for one particular gene having effect on lots of different pieces of the phenotype? Okay. What do we call that? We call that pleiotropy, right? Where we have one gene, lots of different phenotypic effects. So this is an example really of pleiotropy. Okay. Now a slightly more complicated one, the one that's kind of helps us complicated in the structure of it, but it kind of gives us a real view of how maybe a hormone might work and how all this types of work would be the glucocorticoids, which are steroid hormone. They're produced in the adrenal glands, and they're involved primarily in glucose metabolism. And they really have three primary elements that are involved in the regulational process. One is obviously the hormone that comes from the adrenal gland in response to the presence or the need to metabolize glucose. Obviously, there has to be a receptor, which is a protein found in the cell. Okay, the receptor itself has a number of cool parts. It's what's called a zinc finger transcriptional protein. When you see zinc fingers, zinc areas, that often means they're areas that bind up to DNA. So it's a signal for that. And it's a highly modified protein. Obviously, both ends are highly modified. The amine and the carboxyl ends are modified. It has a number of domains that are involved, and one including a dimer formation domain. So remember, we talked about the level of protein structure. And we talked about the fact that you've got primary, secondary, tertiary, the folded structure, and then sometimes you would put two of those or more of those tertiary units to form quaternary structures. And we said when the two were identical, that was called a homodimer. Well, that's in fact what happens here is we get the formation of a homodimer as part of the process of actually moving toward the regulation. So what happens here is the hormone comes in, okay, and it responds to the receptor, and ultimately it ends up finding what's known as a response element. In this case, what they're usually called. What that basically is an area of DNA that it's actually going to attach to. Okay? And so the steps are something like this: the hormone binds to a receptor, and usually at the cell surface. Okay, that imports this complex of the hormone and the piece of the protein moves in the receptors. Once that's inside the cytoplasm, it actually forms now a homodimer, finds another one. That's what this is illustrating up here. This is one side of the protein, the polypeptide that's formed up, and this is the second side, and this is the same thing that's red and blue down here, with this being the DNA. Okay. Once it's formed a homodimer, it's able to be transported into the nucleus. Once inside the nucleus, it complexes up with the response element, okay, which is basically a promoter region, if you will, right? It actually physically attaches with these zinc areas to the DNA itself. And when it does that, it activates these transcription factors. Okay? So it causes transcription to process to start the initial formation of all this big transcriptional complex. So it's a trigger for that. Okay, so it's kind of interesting. You've got a hormone, you've got this receptor. The receptor does, is a protein that does interesting things itself, and then together those are able to move into the nucleus. So it gives you a little example of how maybe a true hormonal response might work. Right? And as I said, we were going to look at transcription factors and talk a little bit about them. So what are transcription factors? Well, factors just means there's something, okay? They could be mostly proteins, but they could also be RNAs. They can be all sorts of things, but the point is they tend to bind to the DNA itself. And once they form this whole transcriptional complex, they greatly increase the amount of transcription that can occur. And we'll see what that means in a minute. They do it just as we saw a moment ago by binding. They have these binding domains, right? So and many of these transcription factors have, once they're in their tertiary structure, and sometimes in a quaternary structure, as you just saw, but in a tertiary structure, they have a domain that will bind, and sometimes those are zinc fingers, sometimes they're other structures. Okay. What's interesting about it is that you can have a single gene, and it can have multiple places. We saw before when we looked at some of when I showed you the promoter region for my P62 gene. I said, oh, by the way, there's TATA, and there's all these other SP sites and things. I showed you a number of different areas that were, ahead of the gene. Those are all binding sites for transcription factors, and they can be different transcription factors. This gene can be turned on by this one activity or a completely different activity. So they can have multiple transcription factors that can control the expression of a single gene. So it gets very, very complex. Okay. It turns out that about 10% of this DNA stuff that we have that we would call the things that get transcribed that we call genes in humans is our transcription factors. So a lot of our DNA is actually involved in producing protein that's going to come back into the nucleus and help with the transcriptional process itself. We invest a lot of our genome and our effort into that. Okay. Now, I mentioned that this 100-fold increase or whatever. That basically occurs because transcription factors can kind of work in a couple of ways. One is they can sit down in the promoter region of a gene by themselves, and in doing so, they generally allows, sort of what's called basal level or low level production of transcription. So they'll just be in the presence of them there, and you get production of the protein. If you need a lot of the protein or you really want to control it, then it gets much more complex and starts to involve things like enhancer regions. Okay, remember we talked about enhancers. We mentioned enhancers and TATA boxes and CAT boxes and those sorts of things, right? Well, these enhancers, when they're present and when they interact with these transcription factors, they vastly increase the amount of production, the rate of transcription, not only the rate, but sometimes the number of transcription that goes on. Okay, so what I want to do is I want to take a look at enhancers and how they actually work. But to do that, we also need to talk about some other elements. Okay, there's things when we think about regulation. Really, we have to think about things that are like silencers as well as things that are called insulators. Okay? These all are pieces of DNA, or they're proteins that function, that recognize pieces of DNA and help control the system. What it turns out is that our genome could be kind of thought of as a schematic for some type of uh, electronic system. I know you're not, most of you are not aware that there was a time before the internet. Before the internet, and then before all the fancy things that we could do with computers, and when growing up, we would, as kids, we would build things, projects for things. One of them, we could build little radios where we could listen to my, um, whatever, music stations, or so if I was lucky, I could pick up stations that were playing my St. Louis Cardinals or something. But we would kind of play around with these things, and we would learn about, we'd build little systems to make speakers or to do various things. And in those, we would learn about things like blocking diodes. Blocking diodes are things like, are basically insulators, amplifiers, which are enhancers. There's all sorts of transistors and things that we've learned about. And it turns out, as I got into genetics and we started understanding more about regulation, you almost go back to the schematic world where you want some stuff going one direction, but you don't want it to go in the other. If it goes this way, you want to amplify it. If it goes this way, you want to reduce the amount. And so we have these structures, it turns out, that kind of mimic this whole process. So let's take a look at them. Let's talk about enhancers. Okay, these are enhancers of these upstream regions, and they can actually be downstream. We'll talk about that in a moment, but they're regions that are separated from the gene, usually most often upstream, that bind to these actuator proteins. Silencers are regions of DNA that respond to these things called repressor proteins, and which basically can shut down the process. And we'll talk about one example of that, and then we'll give another example of what an insulator looks like. Okay, but first let's deal with these enhancers. All right, so again, enhancers are just stretches of DNA that are somewhere away from the gene itself. Okay, generally when they're not active, the gene is inactive itself, although in some cases you do have some basal production. It depends on the gene, but for the most part, if the enhancer is not active, the gene's not active. In order for it to become active, you have to build this complex. Okay, and the complex is quite important because you've got to have this TATA binding proteins, you've got to have all these other proteins that come into play. And once you have this transcription factors in the enhancer, then you're allowed to have RNA pol come in, you're allowed to get transcription, okay? But it doesn't actually look this simple. It's very often something like this, okay? The enhancer is way off up here somewhere. There's literally a folding of the DNA down to help cap this whole process. Remember that we talked about capping this process. There's often looping of the DNA itself to bring the enhancer into contact with the pol. All these things sort of have to happen, and it has to be protein mediated, right? You need to pull all these things together for us to work. So you just think about, here's an enhancer out here. Here's my gene. I've got to bring that enhancer into place and move it into the area. Okay. Well, it turns out it's even more complex than that. Depending on the genes, you can have proteins involved. You can have all sorts of other nuclear level RNAs. Okay, NC means non-coding RNAs, and these can be relatively long or short, but these are RNAs, so these are transcripts that have been transcribed. They've been modified usually, and they maintain themselves in the nucleus itself. Okay, and they become involved in this whole process, both by helping to find the piece that you want, or by forming up. And they can be fairly, non-complex, where they are directly involved in the process, as in Hox clusters, all the way up to these neural activity systems, which are highly complicated with a bunch of cofactors and multiple enhancers and promoters, all interacting with these drugs. And interestingly, it turns out that these RNAs can actually mediate the type of transcription that's going on. Most of the time, and particularly with genes that are, that are going to be protein production genes, you have unidirectional transcription, right? Remember when we showed you what genes look like, and I talked about the fact that sometimes it's this strand, and sometimes it's this, and sometimes they overlap, and they do all that depending on what you go. You only transcribe one way generally. Okay, well that's certainly true for areas where we're going to make message, classic message with caps and tails and all that. But when we're transcribing things like tRNAs, when we're transcribing other nuclear RNAs, things that small nuclear RNAs, actually it can be bidirectional. And when we're going to do a small piece, you can actually go both directions simultaneously. And this is, again, all controlled by this whole mechanism. And very often it's this RNA that controls whether you go bidirectionally or unidirectionally involved. So again, it becomes a very complex sort of problem. Obviously, you could spend an entire semester talking about one or more of these simple models that we have here. Now, what about silencers? Well, just to give you one example of them, silencers are generally, or very often, just areas of DNA where you get something sitting down on top of it that stops the process. Rest is a classic one, okay? What happens with rest is that there's a repressor. It's a transcription factor, okay? It's called that because it in this case stops transcription, but we're regulating transcription. And it serves instead of as an actuator, instead of like you would with an enhancer system. When there's a silencer in place, what happens is generally that silencer is fairly often, well, it's often close to the TATA box. And physically, that transcription factor, the repressor, is big enough to come in and actually block the TATA site. And what that does is it prevents the binding of and building of your complex. So even though you might get a response element coming in trying to form something, if this repressor is in place, you can't get the formation of your transcriptional complex. So certainly can't get your TATA binding protein to come into play, and as a result, you get no transcription. Okay, a good example of that is some of this RES feature. It has a number of splice variants. Okay, we've talked about splice variants before. When you use this exon sometimes, you don't use the others, etc. And these turn up in different organs. Okay, it's a very complex example. I'm not going to try to go through everything about it, but basically for us, in neural cells, RES allows you the transcription to occur. In other types of cells, skin cells and other things, it tends to shut down transcription. So it truly is in that place a silencer or a repressor. Okay. It doesn't shut down everything. Obviously, it's targeted towards certain gene regions. It's highly active during fetal development, and it turns out to be active late in life. Okay, and normally what it does, okay, is that the RES protein sort of prevents, okay, prevents. It is attached to other proteins, and when it's present, it's outside of the nucleus. It doesn't have its normal job. Its normal job is to prevent or to silence. Well, if it's attached in a normal system like this, you get transcription, which is important in things like neural cells because neuronal survival, the dendritic growth, and that sort of thing are controlled by genes that are affected by RES. Okay. Now, what happens is that it sometimes becomes a problem, right? One of the places we see a problem with it in is in, say, Huntington's disease. In Huntington's disease, these cofactors that prevent rest from entering the nuclear region don't work. They've been mutated. Rest is allowed to go in, and rest comes into play. And what happens is you get a shutdown. It blocks production of various types of RNAs that are needed for maintaining these nuclear regions or maintaining these neural systems. And so you end up with a number of problems, one being that simply the transport of some of these proteins that are supposed to be broken up and moved out. They don't get effectively broken up, or they misform, and you get too many of them, and they get stuck on the outside of the dendrites, and they end up blocking transmission in our neural system, which causes problems and all sorts of things. Okay, it tends to be, interestingly, it's downregulated in Alzheimer's individuals, which means that there's a secondary pathway that is a problem. And in fact, it's been shown fairly recently that one of the reasons it's downregulated is that there's this repeat region that's actually found, this internal to particularly Huntington's disease is a good example. There's a tandem repeat. Remember those little repeats that we talked about and how they can grow and shrink? Well, in one form of the early onset Huntington's, there's a large set of this repeat in the middle of the actual gene itself, which means that you get a lot of this protein amino acids put down in the polypeptide that you don't need. Okay, and it makes the protein misfold. Misfolded proteins don't work, and you end up with a problem. Okay. Interesting though, just the opposite happens, and we're not sure why. But in those people that lived, centenarians, the people that live a long age in their 90s into their hundreds, rest is actually highly activated in those individuals. It's highly present, and you're thinking, well, why would what? That seems to be the opposite of what you would want. It's neuronal survival and things, but it's present. So it's a real question of what's going on there. I don't think anybody actually fully understands at this moment the system. But the point for us is that it's this protein. It can go down and sit down on a specific spot of the DNA, can block the transcription directly of some important components. In some cells, that's a good thing. Some cells, it's a bad thing. Depends on what. And there's cellular mechanisms to either optimize its presence or to reduce its presence in the nuclear area of the cell. Okay. All right. What about insulators? This is the last one I want to look at here. Again, I told you this would be a bit of a long lecture. Insulators are pieces of DNA. Okay. They tend to be located between enhancers and the promoters, and basically they sort of stop. And they just prevent the enhancer from going somewhere. It's those of you that know anything about electronics, they're essentially blocking diodes. Okay, they don't allow the system to move past. Okay, and they're important. There's some very common ones that are found in vertebrates. And so what they really do is if you have, say, an enhancer area up here, a hormone comes into play, and you've maybe got 3 genes involved. That hormone is then able to activate this gene here, skipping over this green one in this case, and activating one off down here. And so they protect this green one from being activated by this particular hormone or this particular set of enhancers. Okay, this one then might have its own enhancer, or it might be affected in a different way by another. The book shows a nice little pattern of it like this. It basically is an insulator to prevent these enhancers from hitting the wrong gene. So if you have this enhancer right here, you don't want it jumping over and turn on gene A, you want it to turn on gene B. And so this shows a fairly simple way that it works, but it's an insulator region. And often there's an insulator binding protein here, right? If you remove that protein, then you allow this one enhancer to affect gene A. If you have the protein present, it sets down just like those silencers do, and it prevents kind of movement of that enhancer back and forth. So it's an interesting area again with yet another protein that's produced that has to form here. Okay, a real world example of it, just to finish all this up. IGF. I mentioned that IGF-1 at least being one of the byproducts of this bovine growth hormone that we do have to worry about. This insulin growth factor 2 is common in mammals, and what's interesting about it is that during development, during fetal development. Okay, so this is a growth factor, so it's going to make cells replicate and organs develop and things. Okay, only the allele that we get from dad is active. Okay, mother's allele, the one you get from the maternal side, is inactive. The reason for it is that there is a insulator that is found for this gene. Okay, and that insulator is actually activated. Okay, it's activated by methylation in the male. So during spermatogenesis, there's methylation that goes on that says use this gene over here, don't use this gene over here. Okay, and it's the CCCT factor. It doesn't matter, but that's very often where you see methylations occur. So it's a piece of DNA that's been methylated. Okay, and that methylation allows it to function at, to release the insulation process. In females, it's locked down, it's insulated in the form. And we might remember the term for all this. We've seen it before when we talked about Angelman syndrome, and we talked about some of those other areas. This is genetic imprinting, right? This is one of the ways that we do genetic imprinting is by the use of these insulators and protectors and silencers, for that matter. In some of the cases when we send a signal down through our gametes is by silencing, by putting actually, the male or the female will produce a protein that acts as a repressor. The male will produce it, the female won't, or vice versa, and you end up using that. So that's one of the ways that we actually control which one of these we signal to our children by the production of our gametes, which genes we want to use. Okay, and it's one of the reasons that we can't at the moment just produce clones from human eggs effectively and just get a functional individual, because taking two human eggs and fuse them together, we don't, in this case, we wouldn't have the growth factor activated. So it's one of the areas that we're really trying to study and understand is how regulation of these work and what would need to be done to actually make that function. Okay, someday it will be, and those of us that are male will not be needed anymore, which might be a good thing. I'm not sure. Okay, depending on your opinion about it. Just to point it out, okay, just to kind of take us home on all of this, we're talking about transcriptional control. We've talked about these how things can happen with our nucleosomes and all that. We now have talked about things like silencers, which just basically can act either directly at the DNA level. It turns out they can also act at the nucleosome level. We can have these insulators that protect a form. And just to mention this, and generally enhancers are upstream somewhere from the gene, and they will fold back and make a point. But you can also have what are known as distal enhancers, which are generally enhancer regions that control multiple genes. So not just the one that's next to it, but they have a whole suite of genes that they may activate or deactivate. Usually, in this case, activation. And just to point out one last time, there are downstream enhancers. There are enhancers that actually fold backwards this direction to modify the system, although most enhancers tend to be upstream. We are finding more and more often that there are enhancer areas that are actually downstream. And this enhancer may respond to one input, right? This might be an enhancer that's required all the time, or it might respond to a different one. And this one might even respond to yet a third signal. So, it can really it is a complex system. So you wonder, okay, when we go in and we don't understand why these genes are turned on, okay, what's going on? Can't we just turn that gene off because it's causing this problem, this genetic problem? Well, because we don't sometimes even know what's really turning it on and turning it off, or how it's turned on. That's a very complex thing to have to do. The only way we know that is to go in and start sequencing and things, and then shutting down this enhancer and seeing what, mutating it, see what happens, or putting things in that will increase the enhancer response. So, trying to make these, pharmaceuticals and things that do this is a really complex area, because, you might stop this enhancer and therefore shut this gene down if that's what you want to do, but that enhancer also might be important for another gene down the road. So if you turn it off, you not only lose this one, but you lose the other gene you didn't mean to lose. So it becomes a big response problem that's a really active area and kind of a very, very interesting area, particularly those of you going into pharmacy. It's a growth area that you'll see there. All right, So we've gone through transcription. Now the next lecture will kind of bust through the other forms lower down, because while they can be just as complicated, we're going to kind of just work through them fairly quickly to see, yeah, you could also control it at the maturing of the message. You can obviously control translation as well as maturing of the protein. That'll be in the next lecture.